In today's presentation, LI-COR's technical support scientist, Elizabeth Gordon, will discuss and demonstrate the Insect Respiration Chamber for the LI-6400XT Portable Photosynthesis System. She will also take an actual respiration measurement from a hissing cockroach. In addition to Elizabeth, we also have several panelists with us today to answer your questions. Panelists, please introduce yourselves.
Shannon Lorio, Science and Support Manager.
Eric Price, Applications Analyst.
Alyssa Menzel, National Sales Manager.
Kayla Kemp-Smith, Leaf Coordinator here at LI-COR Biosciences. If you're not already familiar with the LEAF program, this program was designed specifically for primarily undergraduate institutions to obtain research-grade instrumentation for their classroom. This program offers grants or exclusive discounts towards the purchase of photosynthesis instrumentation, soil flux instrumentation, and light measurement instrumentation. If you would like to learn more about the LEAF program, you can certainly visit our website, or you can feel free to contact me directly, either through email at leaf@licor.com, or feel free to contact me by phone. If you do have any questions or have any feedback regarding this webinar, feel free to email us, or you can certainly post on our online forum.
With all that said, I think we're ready to turn it over to Elizabeth to begin her presentation.
Welcome to the webinar, everyone, and thank you for joining us. Today, we're going to discuss insect respiration and using the Insect Respiration Chamber for the LI-6400XT. We'll also have a section with a demonstration of insect respiration, if my insect will cooperate, and of course, I'll answer your questions. So, let's get started. First, let's define what we're talking about. Most of the time, the LI-6400XT is used to measure photosynthesis, which is the first equation on the slide. However, since it's a gas analyzer, it can measure gas moving in either direction, out or in, which means we can also look at respiration of various types, which is the second equation. It could be soil respiration, which is the respiration of roots, microbes, arthropods, and other organisms within the soil. It could be leaf respiration, which is composed of mitochondrial respiration and photorespiration in C3 species. And of course, it can be whole organism respiration, which is what we'll look at today. As a gentle reminder, when we talk about respiration in non-plant organisms, we're talking about the way an organism uses food and oxygen to generate energy for cellular processes. We can talk about either cellular respiration or overall organismal respiration. This involves things like the Krebs cycle and glycolysis, which I won't go into because we've probably all been taught about it at least 5 times since grade 7. The key here is oxygen consumption and carbon dioxide production, which plants then use to make more oxygen for us. And that's where our little many-legged friends come in. Insects in general do not have an oxygen-carrying chemical in their blood. Most insects have a waterproof cuticle unless they live in a moist environment. In that case, the cuticle is permeable to water, and they obtain their oxygen by diffusion across that cuticle. If an insect has a waterproof cuticle, it draws in oxygen through holes in its body known as spiracles, and then pumps the oxygen through a system of increasingly tiny tubes that deliver the oxygen directly to tissues and muscles. They typically have a pair of spiracles for each thoracic and abdominal segment. The same tubes that transport oxygen into the insect body and tissues also remove carbon dioxide. Insects use different methods to release carbon dioxide, including opening the thoracic spiracles to take in oxygen, while exhaling carbon dioxide through the abdominal spiracles. This system is much more efficient than the vertebrate system. Insects deliver much greater volumes of oxygen in proportion to their size. They also deliver oxygen directly to the tissues instead of dissolving it, transporting it, and reconstituting it as most vertebrates do. Here's a short movie showing the spiracle behavior of a grasshopper abdomen. Note that not all the spiracles are open. The spiracles are independently controlled, so the grasshopper can choose to open or close as it likes. You see we've zoomed in a little bit, and you can see a closed spiracle. Still closed. This one is not using that particular spiracle. But now, if we look at a different spiracle, you can see that the spiracle will open shortly. And this, you see, it just opened. This grasshopper is actually using that particular spiracle, but other spiracles are not being used. So let's look at some research that has been done on insect respiration. Because insects take in oxygen through their spiracles, which they open and close as needed, and because they can take in a large store of oxygen at once, they can live a long time without breathing by closing their spiracles and reducing their activity levels. If you look at this graph, closed is when the spiracles are absolutely closed. O is open, and you can see that the volume of CO2 going in and coming out is very high. And then F is flutter, meaning that the spiracles open and close and a little bit of CO2 comes out, but not very much. It's basically a closed state. The insect respiratory system is so efficient that resting insects can stop taking in air even while they're releasing carbon dioxide. This allows them to keep oxygen and carbon dioxide levels in balance. Too much oxygen causes oxidative damage to the insect's tissues, just as too much oxygen can cause oxidative damage to a plant or a vertebrate. And again, you can see the open, closed, and flutter stage of CO2 going in and coming out of this little caterpillar. This particular beetle that you can see a body diagram of is adapted to very dry habitats. And in this case, these authors found that the flow-through respiration measurements showed them it was only using one spiracle for respiration. And because only a single spiracle was open, a very small area of the respiratory passages were actually open to the atmosphere, which meant that they reduced water loss. This dung beetle is native to the desert, so it doesn't want to lose any more water than it absolutely has to. So now that we've discussed a little bit about what makes insect respiration interesting and why we might want to look into it and why we might want to integrate it into our classroom discussions, let's talk about using the LI-6400 XT to measure it. The first thing we have to do is set up the insect respiration chamber. If you have one of the earliest chambers, it might be a little shorter and wider than the one I'll be using, but the principle is the same. There's a replaceable filter paper disc on each of the caps to keep debris from getting into the air lines, and you should replace those when they start to get a little bit dirty and grungy. The cotton you see inside of the chamber can be removed. It's left over from this tube's other use as a chemical column for some of our gas analyzers. Some chemicals do not play well with others, so we separate them. You can also use it to separate insects if you'd like to do that. We need to install this sample cell outlet. The sample cell outlet is the little silver thing that closes off the bottom half of the sample area when there's no chamber attached to the sensor head. You have to remove any leaf chamber you have attached to the sensor head before you can install the sample cell outlet. Make sure you put the leaf chamber exhaust tube onto the sample cell outlet. It's the little L-shaped looking thing next to the gold mirrors, since you will need to match the sample and reference analyzers. If you're going to be going back and forth between measuring plants and measuring insects that might be eating those plants, you don't have to install the sample cell outlet. Instead, you can just make sure that you have your leaf chamber tightly closed with no gaskets and no O-ring leaks. Make sure you check very carefully for leaks or your insect respiration values won't be correct. Insect respiration values are sometimes not very high. We wouldn't want any leaks to make them look larger or smaller than they are. The other thing we need to do, along with installing the sample cell outlet, is blocking the top half of the chamber. You can use either propofilm or adhesive tape, depending on whether you think you'll be opening the chamber with the handle. Propofilm is a clear plastic film, which means that if it falls out of the chamber, finding it again can be a challenge, especially on a lab floor or in the field. I've used bright green lab tape here because it's easier to see it than seeing transparent film or tape, and I wanted to show you exactly where the tape should be placed. It's right over the 3 holes in the chamber manifold and underneath the 3-hole gasket. We also need to install the insect respiration chamber into the cable bundle. It goes in the sample line, as you can see from the black heat shrink on the cable. So first attach the sample line from the sensor head, and then install the chamber between the sensor head and the cable bundle. It will attach to both the reference and the sample air line, but you won't detect any respiration if you attach it to the reference line, so don't do that. Follow the black heat shrink and attach it to that tube. When it's time to add your bug, I recommend unscrewing the tubing from the sensor head or the cable bundle before you unscrew the end of the chamber. It's much easier than trying to do it without unscrewing the tubing. Otherwise, your bevel line tubing gets really twisted up and hard to work with. Now that we've finished installing the hardware, we need to install the software. The insect respiration configuration is built into version 6.2 software, but if you have version 6.1 or 6.0, you'll need to download the configuration from our website. If you need to do that, the link is at the bottom of this slide. The insect respiration configuration is installed just like everything else. In version 6.2, go to the config menu, choose New, then expand Special Configurations and select Insect Respiration. In version 6.0 and 6.1, you'll need to load the configuration by following the instructions from the program you downloaded. Once it's loaded, it will appear in the list of configurations when you start the LI-6400 XT. If you have version 6.2, the LI-6400 XT does most of the work in modifying the configuration, so all you have to do is save it as a new configuration and choose the name. The default name is Insect RD, and that will work very well, but of course you can change it if you want to. You can name it anything you like within reason, as long as you will understand that this is your insect respiration configuration. Once you've installed the configuration with either method on either software version, you'll see that new measurement mode has changed a little bit. Instead of line C being photosynthesis, transpiration, CI, and conductance, as it is when you're doing leaf level measurements, it's now respiration, transpiration in millimoles, and the mass of the insect. You'll also see changes in the environmental control options, but we'll go into that when we do the demo with the insect. And here are a few tips to help you make the environment in your chamber as pleasant as possible for your insect. Happy insects are respiring insects, so we want to keep them happy. First, make sure you control reference CO2, not sample CO2. Second, set the desiccant halfway between scrub and bypass. Third, enter the sample mass in grams so that the LI-6400 can calculate the respiration rate properly. And since the sample chamber is before the sensor head, match before anything is put into the chamber. The light level and the temperature are not as important as they would be in plant measurements, but we do want to make sure we give our insect plenty of CO2 and water. Okay, so now let's see what the little chitinous insect over here is doing. First, let's talk about new measurements mode. You see that we've got respiration, transpiration in millimoles, and mass. And we've also got on menu level 3, here's our mass instead of our leaf area. On menu level 2, you see we have lamp none. There's no lamp by default, but we still see delta CO2, H2O, flow, and relative humidity. And we want to keep the relative humidity relatively high for many bugs. It's not as important as it is in plant measurements, but we still want to make it so that the bug is comfortable. The other thing I want to do is look and see whether we can see some respiration. I see that my respiration rate is 3.6. Let's see how that's changing. So it looks like we did have a point where the bug kind of stopped breathing a little bit or wasn't breathing as much, and now it's breathing a little bit more. Sometimes you'll see a nice, even, regular spike of breath, but it's not always common. It really depends. Since the bugs can breathe discontinuously, they don't have to breathe as we do, constantly breathing in and breathing out. Let's see if I can startle the bug and ask it to do something a little bit different. So I'm going to take a pen and I'm going to tap on the insect respiration chamber. It's not going to hurt the bug in any way, but hopefully we'll see a respiratory response. So just a second, and you'll see my hand. Okay, so I tapped on it and look at the respiration rate. It went from 3 to 10. I have definitely, and now it's going back down again, back down to where it was before. So I definitely startled the bug a little bit, and so we do see that response. There have been lots of different experiments that you can do with this. You could put bugs into the freezer and then for a little while and then take them out again and see how the respiration rates change as they warm up. You could put a bug into the dark and then see if it reacted to the light. You can do any number of things to see how it interacts with the environment. Maybe give it some plant and then see what it does. Plant material, see if it'll eat it and how that changes the respiration rate. And let me try that again just so that you can see. I was hoping that the bug might get into a regular respiration pattern, but it looks like it might not. Sometimes they do, sometimes they don't. This one's not cooperating all that much today. So let me try tapping it one more time and see if we can see another respiratory response. And we did see a respiratory response. We saw not as much of a spike as we did before, but we did see a spike. So it looks like our bug did respond to that. But this isn't a very long demonstration, but I wanted to see that, we can influence environment and then we can see what the insect does. We could watch it a little bit longer and see if it would go back down to a regular respiration rate. But the chances are that it probably won't. It'll probably breathe discontiguously for a while. So now you've seen what it looks like to do an insect respiration measurement with the LI-6400 XT and the Insect Respiration Chamber, and we've talked about a few other applications for the chamber. Does anyone have any questions that have come in during the demo, Abby?
Yes. One question we received was, does it matter if I use dry or fresh weight?
That's a good question, and it doesn't matter, no. As long as you're consistent between whether you choose dry or fresh, it really doesn't matter.
Another question we got was, can I correct my data for mass later?
Yes, you can correct your data for mass. The LI-6400 XT outputs an Excel file. All the formulas are built in, so all you have to do is just change the correct column, and you'll see everything that influences that column, in this case mass, just ripple through the spreadsheet changing.
Oh, here's a great question. Do I still need to match?
Oh, that is a good question. Someone's been paying attention. Good job, whoever asked that one. Yes, you do still need to match, but you can't match with your insect or your fruit or your nut or your soil or whatever in the insect respiration chamber. So always match with an empty insect respiration chamber.
All right. Well, it looks like that's all the time we have available for live questions. If you've submitted a question, our panelists will answer that before we end the webinar. I want to thank Elizabeth for doing a great job presenting on insect respiration with the LI-6400 XT. And attendees, please watch your email. We will notify you when this webinar is available for viewing online. And again, thanks everybody for coming.